Thank you very much for the kind invitation. Can I get the first slide up? Yes. So as we heard, vision starts in the eye. There's a video that we capture by the retina, and it flows through this pathway that Peter introduced to the LGN primary visual cortex, so it radiates through many different cortices. Now, the visual cortex is enormous. I mean, I think around 50% of the cortex we can in human measure visual information. Yet if you ask medically relevant, what is relevant, almost all diseases of loss of vision is coming from the eye, and the unsolved diseases are coming from the retina. Although if we go to Africa, there's still many, many blinding conditions we see, which we could easily fix today. And that's actually the vast majority of blindness is coming from today fixable conditions. Now, within the retina, we differentiate two conceptually different blinding conditions. One, then the blinding condition is coming because of the loss of photoreceptors or dysfunction of photoreceptors. This is around 2/3 of the blindness is coming from mainstream macular degeneration. And the other one, when you lose the optic nerve due to ganglion cell disease, is around 1/3, very significant numbers. As we heard today is that if you lose, from Peter, that if you lose your ganglion cells, there's nothing we can do in the eye. However, if you lose your photoreceptors or photoreceptors causing blindness, then there's much to do with the eye, and this is what I will talk about today. So the retina, it's important to understand, it's a very sophisticated computer. There is one image or there's one video that we acquire, and there are around 30 different movies detected by different types of circuitry in the retina that pulls out different features of visual scheme. In humans, 5 of them are very important for high-resolution vision. So around 5 of these different types of features responsible around 90% of the output of the retina. Now, if you zoom, if you look into the retina, and yes, good. This was already done by Cajal, and indeed we just repeat what Cajal have been seen before. There's an incredible diversity of cell types. So this is really stunning. You can count around 140 different cell types in the retina. Indeed, it was Cajal who came up with the idea of a cell type, which is a morphology that is repeated many times. And it's important to understand that everything starts with the photoreceptors here on the top. We have cones and rods, and then they talk to bipolar cells. Then they talk to ganglion cells from the optic nerve. And much of the computation is by inhibitory neurons, which are a plethora of different cell types. Now, this idea of cell type has been really instrumental of understanding the retina. And as we and others developing gene therapies for a variety of different diseases, turn out to be that the modern concept of medicine in the eye is really cell type. One because of for safety reason, another for efficacy reason. Now, although the structure of the retina, the growth structure of the retina is very highly conserved all the way from fish to human, there have been a very interesting translational dilemma that we and others have been facing, that strictly speaking, most eye diseases are not reproduced in mouse models. And if we develop actually a gene therapy in a mouse, we never reach the clinic. We have spent 10 years, my lab lost 10 years of thinking that's the case. So, and the reason for that is very interesting. There's 2 reasons for that. One is that if you look at the transcriptomes, the gene expression patterns during evolution in the different cell types, retinal cell types massively evolve. So, for example, ganglion cells, you can barely find any difference between. Even when you find, turns out to be that the closest genetically related cell type in the mouse has the lowest resolution and human the highest resolution. The second one, and even more importantly, that in humans, we have a structure called the fovea, which is missing in all mammals except in primates. And very interestingly, when you talk about vision, you only talk about foveal vision. We see patients coming in their fovea is affected in macular degeneration, they come to you and they say blind. Now, we can see patients which have 95% of the retina is lost, except of the fovea, and they don't understand why is there any problem. So when we talk about human vision, it's foveal vision, essentially, because, and this is where high-resolution vision is mediated, and it's massively higher resolution in the fovea in this structure. And I will talk about later, and we look at patients, why is it so important, actually the structure of the fovea. So the diseases we are working on are photoreceptor-based diseases, which is age-related macular degeneration, form called geographic atrophy. It's the end stage of macular degeneration when you lose vision in the center, or inherited retinal degenerations, which is around 280 different genes leading to the dysfunction of photoreceptors and blindness. Now, the therapy that I started to work on actually in 2002, which is quite far away, is relatively simple. Namely, that was a time when people realized that you can find single genes in algae that can make cells light sensitive when you express them, a field called optogenetics. We heard from Ed about it at the beginning of the conference, is to somehow target these photosensors to different cell types in the retina that's strategically important. Now, important thing is that why do we target an optogenetic sensor and not just the opsins we have in our eye? And the reason for that you need around 20 genes to create a photosensation in a photoreceptor, ALG1. So the question is, why are you so better? And all what the 19 others is doing, or even more than 19, is adaptation. So you have an amazing ability to adapt across 10 log units of intensities. And optogenetic channels has around a 2.5, 3 log unit band they can detect. But otherwise, they are just about the same. And what is really difficult is adaptation to see in the cellar and see in a California light. Now, so I talked a bit about the translational dilemma that, you know, in the sort of around the 20 years I've been working on optogenetic visual restoration, we spent around 10 years of developing everything in mice. And we have very good mouse blindness models. And we had students coming in, and we were so good at restoring vision in mouse that some of them didn't believe that they actually were not normal mouse, but just mouse with restored vision. However, none of it actually were able to bring it to the clinic because almost every step of the translational process is specific to the species, including the aspects which I will later talk about. So we had to kind of reinvent everything that we do in the lab, focusing how can we have human models. And I will talk about sequentially that today the entire translational machinery we do is in a postmortem human retina that we can keep alive now for around 20 to 30 weeks. And that's the only way, and then we go to primates. But the human retina is key here. So we use gene therapy vectors. We have heard very nice talk from Viviana. These are AAVs, which consist of basically 3 parts as far as we're concerned. This is a ball, a 25 nanometer diameter protein ball, this is called a capsid, which Viviana engineers quite ingeniously for doing a variety of things. And inside there is a single-stranded DNA, which encodes the sensor, the optogenic sensor, and the region what we call is promoter. And this promoter is really key. It's basically a postal address, which is a DNA, it's around a 500 base per 200 base per DNA segment, which would tell the cell which cell type should it make the sensor. So the cell would enter by its capsid to many cell types, and then by the promoter would define in which cell type would express this sensor. Now, there are 4 different ways of achieving optogenetic visual restoration in the retina. One to target cone photoreceptors. And of course, if the cones lost their outer segments, so they are not light sensitive, this is the best option. because if we target the cones, we basically restore the same kind of visual processing as normally we have. The second best bipolar cells, the second layer, there is a caveat called A2 hemicrene cells. And finally, the worst one, but by far the simplest one, is targeting ganglion cells. So when you target ganglion cells, there is no processing in the retina. There is no features going to the brain. There's only one. Basically, the retina becomes a photoreceptor layer that then talks to the brain. So what we are doing sequentially is going to clinical trials. We're starting with the ganglion cells, what I will talk about today, and then show you how we can learn from patients what is missing when you target ganglion cells, and then go and develop a new therapy now for cones and then hopefully in 2 years we can compare with clinical trials the ability of these people what to see. But I'd like to do walk through how we develop these therapies. This is just one slide to show that the way we develop promoters is that we have single-cell transcriptomes from the human retina, ataxic and single-cell RNA-seq data, from which, using computational approaches, we come up with short DNA segments that could potentially be a promoter. However, then we have a library. By now, we have around 2,000 different promoters. We have to test them. And I think the really, for us, the important moment was to realize, we compared mouse, monkey, and human, is that if you find a wonderful promoter for mouse, this is not going to work in humans, and vice versa. Very little correspondence. A very strong correspondence between primates and humans, indeed. But essentially, we want to treat human patients, so we use a technology that were originally started in Serge Picot's lab in Paris, who could keep alive postmortem retinas for 3 weeks. This is not enough to test gene therapy. We need more. We need 5 weeks. And Arnold Szabo in Budapest, who worked on it for 10 years, basically changing the solution until you get basically a soup that keeps alive human retinas for initially 10 weeks and 14 weeks. By now, we can keep alive for 20 weeks. So we can basically pre-play the entire gene therapy. So you can take, we now tested around 1,000 different vectors with different promoters, and they can very cleanly target, for example, you see 3 examples of cell type, rods, cones, and horizontal cells, but by now we can target many different cell types. One concept I like to emphasize is that we differentiate academic grade promoters and clinical grade promoters. So there's a lot of excitement in academics to start to target cell types, but I would say the absolute vast majority will not be useful in the clinic. In a clinical setting, you want to target 100% of the cell with a uniform, very high expression level. And very often, when you pull out an academic grade promoter, we still, in my institute, work at least 5 years on it until it becomes a clinical grade promoter. Now, with human retinas, let's say we can test 1,000 promoters over 10 years, but still this is a small number. We have tens of thousands of promoters we can predict. And one thing that comes handy is that now we can make retinal organoids that we can see on the, and this is really a development, Magdalena Renner, who has been a postdoc at that time in my lab, who found a procedure and few starting cell types. We can make organoids which are 5-layer just above the human retina. And this was a major change. So the organoid field, the retinal organoid field, you also see some tissue, but was very far away from normal retina. And finding some iPSC lines that form a 5-layer human retina with all the cell types and being light sensitive was a major step forward. And also Magdalena developed the procedure to make masses of retinal organoids. So we do routinely around 20,000 organoids. You can do screens, and this is, we can run screens, for example, finding promoters, and then we reduce at least the numbers then to test in the human retina. So that's the way we think, is that we test thousands of promoters in human retinal organoids, and then we now go to the human retina, or hundreds of at least, maximum or 1,000. So now I'd like to show you two different therapies, one which are already in patients, and that's the one that we started with José Sahel and Serge Picot, Denis Dalcara, and in a small company we started in Paris for Gensai Biologics. We started with José, and that time we did not have the ability to target cone photoreceptors in humans, only in mouse, which did not work in humans. So we decided to go with a simple approach just to test safety basically, and then to some degree to see where are the limitations, what can a human see when you have no retina except a light sensitive layer there. So for this one, at least at that stage, we needed to develop a goggle which has a camera facing outside and a little projector facing towards the eye. And the optogenetic sensor we have been using is Crimson R made by at Botond. And the reason for that was because it's a red-shifted sensor, so we can pump in more light without toxicity. Now, I like to show you two patients, and the details will be very important. So the task of this first patient is to perceive, locate, and touch an object. And each of these are very different tasks. The perceive is say, well, I see something. The locate is which part of the table it is. And the third one is actually the motor action. And each of them is very, very different, as you will see. This is a patient who has lost 58 years old at the time of therapy, lost vision 10 years before. And it has called bare light sensation. That means it can detect where a light is coming from, but no ability to tear apart objects. The second patient is completely blind, so no light perception, 70 years old patient. So let's look at the video. And maybe just to say that this natural condition, there is no goggle, and without the goggle, there's not enough photons to activate the optogenetic channel. That's important. And the task is to perceive and find that notebook in the middle of the table, which is very high contrast.
Au bout d'un certain temps, je ressens qu'il y a une différence d'intensité.
Yeah, so this is the light perception part.
Mais sur la table, je ne vois pas.
This is so it cannot detect the object. Then we put on the goggles, and now it's very important. What you will see is that you see what the camera sees on the computer down there on the Apple computer. So the patient is scanning. You can see that. And at one point, the object is on the camera. And exactly that's the time that the person forms.
Et alors, c'est où? C'est...
Pardon, c'est sur la droite.
That's a localization. And that's a motor action. So what did we see? So the first thing to explain to you that the ganglions has only photosensitivity in a very small area around the fovea. So it's a tunnel vision, so you have to scan. So most of the time, this patient is scanning until it hits the boundary of the object. But when it hits the boundary object, what is truly remarkable is that the motor action is incredibly precise. I will show you another example. Then it's kind of unreally precise. So it takes a long time to see where it is. But motor memory is incredibly strong even after 10 years. To see an extreme case of that motor memory, I show you another patient, now 17 years old, zero light perception. It's this movie, when she has to count pens. You can see again the camera on the pens, and very beautifully, she's actually much better than the man before. She counts the three pens. 1, 2, 3.
Et le bord de la table du fond est où?
And then detects the edge of the table, right?
No.
And now check the motor action.
Parfait.
So what we see is that first of all, there are differences patient to patient, but you can see there's very good object recognition. Actually, learning and rehabilitation makes a huge difference here. Now, there's no face recognition, there's no reading, and I'll get to that. that will be why we develop a second therapy. The sensation is different from normal vision. Very often, when there is an object, they talk about vibration, which we believe is because how we encode the stimulus. In the way we encode the stimulus, that we, in the camera, we have a change detector. So there's only light going to the retina, and there's a change of intensity, and that is to reduce the amount of light to the retina. So when there are small, small movements of the head and the eye, and then when you get close to a boundary, basically they're flashing optogenetic flashes, and that's what we believe the vibration is for. The dimensional learning is the key component. So, this is now consistent across 5 patients we have seen with different amounts of vision. The key question for us is that where we want to really go is we need face recognition and reading. That's where humans consider vision. Now, why are we missing? So the first point I like to make is that there is no high resolution vision. The patient scans and concludes about objects. Why is there no high resolution vision? And by now we really understand it very well. And this is shown in that slide. So ganglion cells and bipolar cells of the fovea has a very peculiar organization. There is no ganglion cell in the center of the fovea, and that is because the light is coming from the ganglion cell layer, and to have high resolution vision, basically during evolution, ganglion cells were shuffled on the side. So they and the bipolar cells are on a torus indeed around. So what we are doing, that if you have you can beautifully see this torus actually in a monkey injection here, very similar to what you would get in humans, where the ganglion cells here are on this ring or torus. So if you have a light sensor on a torus, there essentially, if you look at the face, there's nothing in the middle of the face because there's no ganglion cell there. And the second one, a photoreceptor in the center corresponds to a ganglion cell somewhere in the torus. So if you excite a ganglion cell, it would report about something in the middle to the brain, because that's how normally it does. So you get a major distortion. In turns out there are 2 options. The one is that you use technology to pre-distort the image, but that has to be done in the micron scale. And we are working with Valentina and Jose in these patients, actually, that if we I have an algorithm how to do that based on the anatomy of the connectivity. But a much easier way is to go for cone photoreceptors, which are on a mosaic, and therefore you don't have to pre-distort anything. So there are 2 major questions that can we develop this and how good it is. Second, are there patients with remaining cones in the center? So this is what I address now. So this therapy we are developing in my institute with Henry Scholl and Nancy George, which goes back to a very early publication from the lab in 2010, but at that time, we didn't have the right technology. So over the last 10 years, finally, we found a promoter now screening in the human retina, with which we can target 80 to 100% of the cones in a human retina, in anywhere in the retina, in the fovea, as well as in the periphery. So just to appreciate how much, this is around 500 human retina, how we optimize, how much optimization need to get here. And then we have to ask the question, do we find function in the human retina? So what happens is that in the human retina that on the very first day we get a donation, a post-mortem human retina, this retina is still light sensitive. So what you can do is that you can put that retina on a multi-electrode array and record the output of these 5 main types of ganglion cells. And this is how we do. Here's a multi-electrode array with around 30,000 electrodes made in ETH by Andreas Hirlemann. We put down the human retina. This is actually usually around 5 minutes after donation. The retina is in an oxygenated chamber. And you can see that we kind of flicker light, and 1,000 electrodes is recording now from ganglion cells. And this is coming from 5 different types of ganglion cells. Here are the 5 types. If you show a step of light, you can see the so-called on-parasol, midget, on-off ganglion cells, off-parasol, off-midget ganglion cells. The name doesn't matter. What's important that each of these respond very differently to the same input. Now what you can do is that you have the retina, you know the human retina is responding, and now you put in culture. So you culture it for 5 weeks. The retina loses light sensitivity in the first 3 days, but now we add the optogenetic therapy to it. So now the cones become sensitive, and now we ask again, how do the retinal output reflect now the optogenic vision, how this compares? And this comes a really, really beauty, that it's close to identical. So again, we tested around 20 different sensors, and one of them is almost gives identical output than of the normal human retina. Very different from ganglion cell-based optogenetics, where every ganglion cell is the same. Now, the human retina is actually computing the same as it normally should be. So now, the last point, the last 2 slides, I'm asking, are the patients with remaining cones? And the reason it was important, because in ophthalmology, the idea was a so-called structure-function correlation, namely that if the patient would be blind, there would be no cone. Except if you ask ophthalmologists why do they think so, they were kind of failing to give you evidence, because normally in a clinical setting, you don't do an OCT recording from blind patients because nobody pays for that, number one. And the second one is the technology was, these have usually vibration, these patients, eye vibrations, and the technology up to very recently were not good enough to get high resolution OCT, optical coherence tomography, when you look at a presence of cone. So what we did, my institute conducted an international study, 18 different countries, around 446 eyes around the globe, going to blind patients with retinitis pigmentosa and of geographic atrophy, and look at these correlations between vision and remaining cone. And what we stunningly find, so this is an important plot, where this RFE means the cone mass in the center of the human retina in a patient. And what you see on the y-axis is the level of vision. The higher this number, unfortunately, ophthalmologists are decided to go this way, the higher the number, the worse is vision. So you can see the top, the completely blind patient, yet the cone mass, there are many, many patients, around 62% of the patients who have close to normal level of cones in the fovea. This is not true actually in the periphery, but in the fovea. So that tells us that we will not be able to target all the patients, but around 62% of the patients. And the question, what will be the remaining one? What we are hoping is that to some degree, ganglion cell-based optogenetics will help, and then we will try a later clinical trial, bipolar-based optogenetics. So this is yet in preclinical phase. We are gearing up for a clinical trial for cone-based optogenetics. But what we hope is that we can get to the regime of going more than the object recognition towards learning what is the highest resolution vision we can achieve. We do not know, and this we only learn from patients. But I think that when we learn this probably will be the limit what we can achieve. Now, the very last slide I like to make a note is that this was sort of preclinical development, but very often we believe that we do basic science that helps translation or clinics. But I like to show you that sometimes you do translation and that massively helps basic science. Namely, I told you that we can have a human retina in a dish that is identically processing 5 weeks post-mortem, and we can keep it another 15 weeks today. So that means that we have in our hand the human brain part that we can study with the same resolution, same techniques, 2-photon imaging, viral targeting of cell types, recording of connectivity, and now very recently in the last months, we were able to induce genetic diseases using CRISPR engineering to the same human cells. So we can create disease models actually, and we can, this is human retinas, we can study how they compute, how disease evolve. So this is kind of the feedback from clinical development to basic science, that we now can study human brain parts at an immense resolution and technical ability. And this I like to finish, and thanks many people in my lab and past lab members, and especially José Sahel, with whom I collaborate since many years, and Hendrik Scholl and Bence George, with whom we developed the cone-targeted optogenetic. Thank you very much for your attention.